Morning, everybody. We are going to start with the presentation of the first seminar in that take part of the activities of the psychology, cognitive psychology lab, our lab, as you know. And this is also part of our lessons in introduction of cognitive psychology. So the idea of this presentation is to finish the first block related with perception and sensation. And today we have a special guest, Silvia Savazzi. Silvia Savazzi is professor in the University of Verona in the Department of Neuroscience, Biomedicine, and Movement Science. And she is head of the PandA lab, that means Perceptual and Awareness lab. She's working actually in topics related only with sensation awareness, and she's also working with different kind of patients, as hemianoptic patients, such as hemianoptic patients, patients with neglect, and other different type of patients. We have a long collaboration for 6 years. I was working together with her team in Italy, and she has a lot of publications, and as I told you, she's the head of this lab, and I think that today we can learn a lot about perception and awareness with Silvia Savazzi. So welcome, Silvia. Welcome to our first seminar. You are the first one, and the scenario is yours.
Okay. Thank you, Javi, for the invitation and also for the presentation. Everybody can hear me also. Okay. Javi told me that you may come from different fields, so I'm trying to be as clear as possible. Of course, if you have any question, do not hesitate to raise your hand, and I will stop and you can ask me a question, okay? As the title says, what I'm going to talk is perceptual awareness in the blind visual field. Specifically about subjective reports and the neural markers we can find using specific techniques. What is perceptual awareness? Basically, we don't really have a single definition of awareness. There are many, but to give you an idea what awareness is, think if it works, okay, about this question you may have heard about. What is like to be a bat? So basically, every organism, also human beings, if they are conscious, it means that they have conscious mental states. So basically, being a bat is feeling like a bat, being like a bat, having perceptual experiences a bat have. So at the end is all about this word qualia, which is very used by philosophers, meaning the subjective character of the experience. So when you experience, you see something, the features you experience about this something is your conscious mental state. So when you see an apple, you can feel the colors, the shape, the size of the apple. If you take an apple in your hands, you feel the weight of this apple, and blah blah blah. So basically, every subjective experience you have, and you can also report about, is a qualia. So is the content of your conscious state. Okay. I guess that you know about a bit about the physiology of vision. Just to recap, because I'm talking about visual sensations and visual awareness, just to give you an overview, but I'm quite sure you all have these notions. Basically, when you see something, if you see something in your right visual field, what happens is that this something goes to the left part of your brain, and vice versa. When you see something here, it is processed by the contralateral part of your brain. You may also know about the type of neurons we have and the size of the representation of in your brain of the visual field, meaning that the fovea, the central portion of your visual field, is overrepresented in your brain, and that's why you see details in the center of your visual field, and you don't see much details in the periphery here because you have less neurons processing these informations. This is important because it may tell you something about how we see things by processing these stimuli in your in your brain, basically. But there's not only the primary visual cortex, this area here, that processes visual stimuli. Basically, you have that all the back of your brain here in orange can process visual fields, can process visual stimuli. This is in the center of the brain. This is the lateral view, but all this portion can be used to see something. You may also know that these visual areas can be divided in basically two main streams. The ventral stream coming from the visual cortex to the temporal lobe, and the dorsal stream coming again from the occipital area to the parietal areas. And there are two streams with two different purposes. Basically, when you perceive something, so you have to say which orientation a stimulus have, you use this ventral stream to do this. Instead, when you have to grasp something, let's say the apple I talked to you about before, to take it into your hands, you use this sort of dorsal stream. So they are two streams, but they both, for different reasons, they both process visual stimuli. And indeed, if you look at the visual map, here is the parietal areas, the occipital areas, and here you go to the temporal lobe. You see that this visual field can be mapped on your brain up to the this area here, okay? So all these areas colored here can process visual stimuli. So not only the occipital cortex and the primary visual cortex, but you also have temporal cortex and parietal cortex. So a lot of our brain can be of help for visual stimuli. What happens, and this comes neuropsychology, when you have a damage in your brain? Basically, if these visual areas I showed you before, coming from the eye to the primary visual cortex, are so well represented here, you can also predict with a certain accuracy if you have a lesion in one specific part of this pathway, which visual field defect you will have. So basically, if you have a damage, for instance, here, you will be blind for all what happens, since this is the left hemisphere, you will be blind for all happens in the right visual field. And basically, we study patients with a lesion. The post-chiasmatic here is the chiasma, in which you have that all the neurons are processing stimuli which are presented contralaterally. And how do we assess these visual problems? Basically, by using this apparatus you see here, which projects a light in this thing here, and you ask the participant the to say whether or not they see the light. And the result of this testing, which is a perimetry, is represented here. Here you have one eye, the other eye. Black portions are those portions in which you don't see anything, almost anything, or you see something, okay? So basically, using this perimeter and projecting lights in different locations in your visual field, you can assess whether a patient can see and how much this patient can see, okay? And we use this test to say, okay, if I present something here, I may be almost sure that the participant, the patient here, will not see anything, okay? Because this is a blind visual field. However, this is not always the case. Not all cases in which you have a perimeter saying that you don't see anything, you also have no perceptual experiences at all. And these experiences can be of two types. They may be unconscious, but have an effect on the conscious or on your behavior, or being conscious by themselves. To give you an example of the first case, so stimuli presented in the blind field, which have an effect on the what is presented in the good field. Let's see about these figures here. I guess you see a triangle here, and you don't see a triangle here, right? And this is because this creates these three, let's say, Pac-Mans, create a configuration, so a gestalt, allowing you to perceive a triangle. Let me say that this falls into your blind visual field. So you don't see this Pac-Man here. So in all these cases, the only thing you see is these two Pac-Mans here. But what's different in the patient's response is the number of times patient report a triangle. If you are in this condition, mostly the patient report a triangle, but they don't see this Pac-Man. Here, instead, they do not report a triangle because there's no triangle, right? However, they, in this case, and in also this case, they only see these two Pac-Mans here. So what changes is that what is presented in the blind visual field have an effect on your perception. So you perceive something despite the fact that you didn't see this specific Pac-Man here. And the same is for this figure here. You report an hexagon or a pentagon depending on what's present in your blind visual field. So here is an effect of something you don't see that can be detected on something you see, okay? Another quite old experiment is this. You present this thing here, which is a like a C here, and you don't see anything here because you are blind in this portion of the visual field. But if I present to you something like this, so two segments, one you see and the other you don't see, what you report is these elements, even if you didn't see the element, which you don't do, of course, in this condition in which you are presented with only something in your intact visual field. So again, this example tells you that what's in your blind field have an effect on what you perceive. However, in this case, you are not conscious of what is in the blind visual field. So what is in the blind field remains unconscious. You don't say, oh, I see these two elements. You say, this is what I feel there, but I only see this one, okay? Another thing, and so in this case, you don't have qualia, you don't have a visual experience in your blind visual field. But there are other cases. Here is a collection of examples with different patients in which they have presented several types of stimuli in their blind visual field, and they reported something under certain circumstances. For instance, here, vigorously moving stimuli, they report consciously that they saw something. So let's say a flash of light, a pinpoint of light, a dark shadow, changing the names, but what they report is the conscious content of something. So these stimuli come out from the blind field, because should be blind, this field, right? So they should not have any visual experience because this field is blind. However, what happens is that using certain types of stimuli, this patient can consciously report something which is presented in their blind field. So the question is, how can I assess whether a patient is conscious or not? Each time I ask a patient to report something, you know that we have the problem that the patient has to be certain, almost certain, sure, can decide whether or not they saw something. So basically, the way we ask participants, the patients here, to report their sensation may change the sensations they report. They may, for instance, be prone to say, no, I didn't see anything, if they are not sure about that anything. Or they may have a criterion which is, I saw something, but if I compare this something to what I normally see in my good field, this is not seeing. So I may say, I better say, no, I didn't see anything. And this is the logic of the subjective reports and the importance of the subjective reports. So basically, what we tried to do is by asking a patient with, here you see a very small lesion in the primary visual cortex, and this is her visual field, to see whether the way we ask participants to report the content of their consciousness may change the results. So here you have a typical experiment we have in visual thresholds or visual perception. You have a stimulus, and you have to say whether this stimulus has one feature or another. So here you have to discriminate something. And after that, you have to say, I saw something, I didn't see anything. So in a dichotomous way, or in another experiment, you are asked to report in a more graded manner whether you saw something and the quality of this something. In this experiment, we use different features, so to be sure that we assess this, if there is a difference here in using different characteristics of the stimulus: orientation, color, contrast, and motion. And basically, when you here you have the percentage of detections when the participants guessed. So they say, I didn't see anything, or I saw something. And what you see that in most of the cases, with the only exception of this, mostly 100%, 90%, the patient said, no, I didn't see anything. And here in the discrimination, because she was forced to say vertical, horizontal, red, or green, she was always, you see the asterisks here, she was almost always over chance, meaning that she didn't see anything, but when she didn't see anything, she was anyway accurate. And this behavior is called blindside. So I have no visual experience, but my behavior is over chance, which chance here 50% accuracy. However, what happens if I ask the participant to use a more graded scale and not just yes or no? We did this by asking, using this scale, for instance, you have no experience. You can say, I didn't see anything. I had a brief glimpse. So I was a sort of flash, but I couldn't say more than that. It was almost clear. And there's another, but she never used it. It was a clear perception. So what you see here is that detection changed a lot from the yes or no and this graded scale. If, for instance, here, look at the orientation. It's almost all cases she said, I didn't see anything. In contrast, here, giving her the possibility to say, yes, I saw something, but it's not good as in my good visual field, she started to say, to use also this portion of the scale, okay? And the most interesting case is the contrast in which you have something which is seen here, and half of the trials didn't see anything. However, here, she almost never reported no experience. So one possibility here is that she was perceiving something, but it was so low level that she didn't feel to say yes. However, here, giving her the possibility to say brief glimpse, she used this category. So saying that maybe sometimes if we use a yes or no response, we may find lots of no responses which are not no responses, are conscious but degraded, not good perception, conscious perception. So here, what I wanted to stress is the importance of asking the right question to assess in the good way whether or not we are conscious of something. Going to the neural correlates, I may ask, can I see correlates in my brain that go together my responses? Basically, you can do this by running exactly the same experiment. For instance, here I show you healthy participants, but I'm going to show you also patients, adding EEG recording. I guess that everybody knows what EEG is. If you don't, raise your hand and I will say something more. No, everybody? Okay. EEG is electroencephalography. You basically record the electrical activity in your brain. And this electrical activity, which can be recorded in continuous, can be time locked to the stimulus. If you time lock your EEG, your activity in the brain, to the stimulus, what you get are event-related potentials, which are the way, the language your brain uses for making you perceive something, talk, and blah, blah, blah. All your cognitive functions can be translated into event-related potentials, so which are potential electrical activity induced by the presentation of the stimulus. And I'm going to show you something more in the next slide. Here you have this, exactly the same stimulus as before, the same question, a discrimination task, and again, the same awareness scale in which I ask no experience, brief glimpse, almost clear, and clear. In EEG literature, you have, this is a event-related potential, which is a wave and it is subsequent components one after the other. Here is the 0 point in when the stimulus appears, and these are the potentials, so the electrical activity in your brain that correlate with different processes which are in time is doing. For instance, in the first are thought to be more related to the perceptual component, so the sensation, the processing of visual features, and blah, blah, blah. The later, instead, components are more cognitive components, which are influenced by attention, by language, also emotion. So basically, you can tap what is going on in your brain while you are seeing a stimulus. So these brain potentials have been thought to be different when you have that the participants say, I see something or I didn't see anything. And the two components are one very early here and one late here. But you see that these two are different, and also these two are different. If you subtract the two, you get this difference wave in which you have that this corresponds to this difference and this wave corresponds to this difference. And they are thought to be related to two different processes of the visual awareness. Mainly, the early one is thought to be related to the content of your perception. So seeing a red or a green apple, and here instead the later component is thought to be related to what you are doing with this information. So basically, I have to respond to a stimulus. So here you have the decision of responding, doing the response, and blah, blah, blah. So here is more related to the perceptual content, and here is post-perceptual, more related to decision and response. And our question was, can I say same waveforms changing, and which are changing depending on the quality of your perception? So basically, what we did is exactly the same experiment and asked the participant, instead of saying yes or no, to say no experience, brief glimpse, almost clear, and clear. Here you see, which is quite obvious, that increasing the quality of your perception, you also increase the correct responses. You are more accurate if you see the stimulus clearly, okay? But what happens is that this behavior is also can be also seen here. So you see that increasing the quality, you also have that your components will become bigger and bigger, right? Here, the higher one, so the clear experience is the highest here and also the highest here. So not only these components can distinguish whether you see something or you don't see anything, but also they can distinguish the quality of your consciousness. So how good is your perception? And even more interestingly, I think, is when you go to see where in the brain this might happen. I must say that EEG is not that good in saying where things happen in your brain, but the likeliest location when you can see the first component is in the temporal lobe, which is also in line with the idea that when you have to perceive something, you are using your temporal lobe, as I showed you in the introduction, right? While when you have to respond and make a decision, mostly the central portion of your brain is doing this. So basically, also topography of these two components tells us that they are doing different component of your perception. Here is the content of the perception. Here is the decision you are making on your perception. What happens if I study patients with visual field defects? Here you see the black portion is again where the participant don't see anything. So what we did is to present the stimuli in here. But you know, for instance, this is the patient you saw before. You know that they may have some conscious experience also in their blind visual field. But we asked them to run exactly the same experiment. And so again, the stimulus, they have to say whether it is lighter or darker than the background, and again, to assess the quality of their perception. Here you see the proportion of the responses along this scale, and what you see is that most of the responses are almost clear and brief glimpse, together with I didn't see anything. As much as for the controls, what you see is that increasing the quality, you also are more accurate. But what happens in their brain actually? This is the first component, and here you have, in general, what we do is to take the topography. Here is all these dots are your electrodes in your EEG. This is the electrical activity, again, of your brain. The blue means negative and red means positive. Since this is a visual awareness negativity, so it's a negative component, what you have to look at is the blue portions. And what you see is that increasing the quality, also this blue portion increases, okay? And you can see again in the occipitotemporal electrodes, you have this significant difference in the three different levels of your perception by using neural markers, actually, of this content of your perception. These patients didn't show instead this late positivity, so that very big component in later in time. So you don't see any significant here data by comparing the interior. We also run tests for single, but again, no clear topography, no clear results. So I better say that they can process visual stimuli in different ways, and this may be reflected by this component, the visual awareness negativity. But when they report this, their decision is not very strong to make the three component, the three conditions very different. So, and making us to see a clear response in EEG. The last thing I want to say is that you saw visual stimuli presented in the visual field, but you can also create a visual stimulus by using transcranial magnetic stimulation, which is, as the name says, a stimulator which uses magnetic fields to induce an electrical field in your brain, so by changing the state of your brain. Basically, when you use this coil you place this coil on your head, what happens is that you are activating or deactivating, depending on the protocol, your neurons. So if I put this on the motor area, the area that controls your hand, you will have an hand twitch. So basically, you will move your hand, by using this coil. Of course, we are not using motor stimuli, we are using visual stimuli. So, but the logic is exactly the same. When you stimulate the occipital cortex here with this coil, what you have is that the participant have a conscious experience of a visual stimulus, which is created by the activation of the visual areas in the brain. And they can be of different shapes, even colors. They can also move depending on the area you are stimulating. So basically, I can directly, without any visual stimulus outside, I can create a visual stimulus, which is not, of course, an hallucination, because you know that it's not real, but you clearly see a flash of light, okay? And what happens when I stimulate different portions of your brain? Do you have same perceptions subserved by the same areas or not? So in this last experiment, one behavioral and one together with EEG, we used, again, visual areas. As I said, you stimulate the occipital cortex and you see a flash of light. But you also have that also this area is visually responsive. So what happens if I stimulate here? What happens is that you can increasing the intensity of your stimulation, like for normal threshold assessment for visual stimuli, you have that increasing the power actually of your stimulation. You also increase the likelihood, the proportion of your yes responses. I saw something. But these two locations here produce different threshold functions. So here you need less intensity to get to the threshold to see a phosphine. If you stimulate here, you need to go to a higher intensity to obtain the same proportion of yes responses. And here you also can see that each color represent a participant, not the color having seen by the participants. So you see, for instance, that this seems to be bigger than this. They also increase by increasing the intensity, but they are different from one side and the other side, occipital and parietal. What again happen if I stimulate the occipital cortex of a patient or the parietal cortex of a patient with a lesion? Here you have the same patient you saw before, and this is a new one with a clear hemianopia. And what happens is that if you run fMRI, you see that there's no activity in the lesion, which is here and here. So what happens here, if I send a stimulus here, I don't see activity in their lesion, saying that the visual cortex is not responsive. But however, if I stimulate a portion which is responsive for visual stimuli, but it's outside their lesion, you see phosphines. They also increase in size by increasing the power of your stimulation, so they can be induced to see, consciously see a stimulus by stimulating a portion of their brain in the damaged hemisphere. And they saw something in their blind hemisphere, right? Again, here you see their threshold, but it's exactly the same as before. They can draw a line like the healthy participants, but I guess that the most interesting thing is when we added also EEG. So not only we show that I can induce a visual stimulus in your blind visual field, by stimulating a portion of your brain, but also looking for where and how this happens, right? And again, by adding the EEG, what I can see is that basically when I stimulate here, I only see by observing the behavior what is going on. Instead, if I add EEG, for instance, here I see that if I stimulate here, after a while, this activity will go to the other side. So what I can do by adding EEG is also to look at the circuits in your brain which are responsible for your behavior, which without EEG I cannot say. So basically, the experiment goes exactly in the same manner as the others. You have that you are stimulating two different portions. Here you have healthy participants, and you see whether or not they perceive a stimulus. But here you also have EEG that tells you almost how this is processed. So basically, again, you have the same threshold, which is higher for here than here. So you need more intensity here than here, which is predictable by the activity you can see in fMRI, which are different in these two brain locations. And here you have again the waves you saw before. Here, instead of being triggered by the stimulus, here the zero is no more the external stimulus, but it's the TMS pulse you delivered to the brain, and you see how the activity is modulated by the TMS pulse. What we saw is that, again, you have not necessary to see all of this, but look at this area here. When I stimulate the occipital cortex, what I see is more or less the same activity in the same location as for the actual stimuli, the real stimuli. So again, I have that occipital stimulation here is transferred to the parietal, the temporal areas here, like for the visual stimuli. So actually, we are finding more or less the same thing that we found in the real stimuli. So is the temporal cortex that can distinguish between perceiving and not perceiving something. And here again, we see the same thing. But what happens for the parietal areas? When I see a stimulus which is outside, all my brain is activated. So both the occipital, the temporal, and the parietal. Here instead, what I can do is to selectively stimulate this portion and see what happens, which is the role of this portion of the brain in perceiving something which is induced by this. And here we have a different picture showing that it's no more the temporal cortex, but it's the parietal cortex that can code the presence of absence of consciousness. We, of course, did the same with the patient you saw before, which I can induce phosphines by stimulating this area, and I want to see what happens in her brain by using EEG. And again, what we found is that it's the temporal, the parietal areas that can distinguish whether the patient can perceive or cannot perceive. So by putting together all these pieces of evidence, what I would say is that first, at the subjective reports level, I would say that if you want to assess in the good manner whether a patient is conscious or not, you may better think of using graded measures instead of yes or no responses, because you may see that the patient say that they saw something which cannot emerge here using a yes or no responses. The other thing is that real stimuli, or also in the last case, phosphines, can be perceived in the blind field of immunopic patients with a lesion in their primary visual cortex, implying that V1 primary visual cortex is not essential for conscious awareness, but you have different locations in the in your brain that can generate the neural code for conscious awareness. So basically, you have that conscious awareness is an early activity of your brain, which is in the back of your brain, and there's no one single place which have access to awareness. There are different areas that can contribute for the emergence of awareness. And with this, I can, of course, I have, of course, to thank all the people who helped me getting these results, the funding agency, because without money, we do nothing, and all of you for your attention. Thank you.
So, follow the questions. But I think that we can start with the students. That is the aim of this kind of activity. So, questions? Was clear? Bueno. Hola. Bueno.
Okay, so can you listen to me?
Not very loud, but yes.
Can you listen to me now?
Yeah, much better.
All right. So at the beginning of the presentation, you gave the example from Thomas Nagel, the what it's like to be a bat. I was wondering if you can, if we can describe perception in animals just as we describe it with humans, that we need some gradients to realize when we are aware of something or not, or if we just perceive something but it's not completely conscious. I was wondering, would it be like the same thing with animals?
Yes, there's a very huge debate about consciousness in other species. If you have a cat or a dog, you know that they are conscious because you see their behavior, right? So basically, if consciousness is a matter of complexity of our brain, I would expect that mammals will have consciousness. Not as high probably as hominids, so also monkeys, but higher than birds, because if it is related to the complexity of your brain, I would say that you have graded shades of consciousness in every animal. Indeed, we say that they are sentient. When we talk about mammals, we say that they are sentient animals. So meaning that they, for instance, I would say that fear or pain is ubiquitous in animals because it has to be. You have to react to something that is painful, right? And you have somehow to be conscious of that something. Maybe other, I don't know, thoughts, very complicated thoughts can be conscious only in humans because of the complexity of our brain. but I would say that all the animals have consciousness.
Yeah, it's because I think like with human beings, because I don't know, it's a little more complicated. Like that's my intuition. Because when you are aware of something and when you report feeling something, is that you perceive yourself feeling that something, right?
Yes.
And
Yes.
with animals, I wonder if they perceive that they are perceiving something. I don't know if that will be like another level of reporting.
Yes. Well, you really don't need language to be conscious. You don't need to think about your perceptions, for instance, because when you are running very fast in a dangerous street, you are not thinking, right? But you are perfectly conscious of the danger of what you have to do, and blah blah blah. So you don't really have to think about what you are conscious of, because consciousness is another thing. Is related to attention, is related to language, but is not language and is not attention. So an animal that feels pain and you see their behavior, or they feel guilty. If you have a dog, you see the face of a guilty dog. You see that they are conscious of that they did something wrong by their behavior. They don't really need to say, "Oh, I know I did something wrong," right? They have it the face.
I completely agree. I just see that with humans, it just, I don't know, it just makes it more complicated, at least for me, but I completely agree that you don't need to be, you don't need language to be aware of something.
Exactly.
So you can try to formulate the questions and we can translate. Also, we have a translator and Alessandra. Other question? We have another question, Silvia.
Hi.
Hi.
Sorry if my English isn't very good. I have to ask if there is a kind of different results in making different tasks, because as long as I have seen in the presentation, the main task was to detect a spot, right?
Yes.
So, for example, I'm wondering if we can find similar results in, I don't know, movimiento, ¿cómo se llama?
Movement.
Sorry, in movement perception or size or something like that.
Yeah, sure. Basically, for each feature you can think about, you can create a threshold. So, for instance, instead of because I always said something which is related to more to detection, but you can also have the a type of threshold which is called discriminative threshold. So you have different movements or different sizes, and you have to say whether they are the same size or different size. And it's more complicated to run with respect to this, but I would say that in the discrimination you may see something different than in the detection, because detection is easier. You don't have to process the feature. You only have to say whether the stimulus is present or absent, regardless of the feature. Instead, when you go to the feature, is adding another content to your consciousness. So you may need to get to the higher level of the graded scales I showed you. So it could be possible that you have different waveforms, different distribution of bands in the EEG, or also locations. But this is a good question.
I have a question regarding the study in the myopic patients. Because, okay, the history of the myopic patients is early. I mean, with the study by Wayne-Krantz and colleagues, and they found blind sight. And now we have a lot of studies discussing the awareness or not in the blind field. So now I know that because we are working together also in this field. Now I cannot imagine to study the blind field of patients without asking about the consciousness in the blind field. So what do you think about that? Because, I mean, when you have a patient with a ophthalmologic problem, I mean, they cannot see because they have the problem in the eye. So you maybe are sure that nothing is happening in the visual cortex. But when you have a lesion in the visual cortex, you're not sure what is not happening or what is happening in the visual cortex. So maybe you can find a lot of different kind of phenomenon in the blind field that we are not actually studying. I mean, because we suppose and we expect that they are really blind, and we are presenting stimuli there, and we say they are blind, they cannot see that. What do you think about that?
Well, of course I agree with you. That it is absolutely important, because there are lots of papers out there saying, okay, I have this patient with the blind field. I didn't ask them nothing, and I assume that they don't see anything. I run the experiment and then say, okay, they are better than chance level, so they have blind sight. But if you don't have, or you are not sure, or cannot be at least quite sure, that each trials, each time you presented the stimulus, the patient didn't see the stimulus, you cannot really say that it was unconscious just because of the visual field. You always have to ask participants to say trial by trial, time by time, whether they saw something or not. I would say using the a more graded, but at least a yes or no response. You may see something that perfectly correct, but you have to ask the patients, because if the damage is in your brain, you have so many alternative streams in which the activity can reach other portion of the brain, which can have the ability to process these stimuli outside V1. So if you have a lesion that involves V1, there are other ways for the stimuli to reach other areas. So you always have to do it.
Thank you. Yeah. Okay.
Hi, Silvia.
Hi.
Thank you for your presentation.
Thank you.
Maybe, I want to know your opinion from your own conclusions. You will say that there is a hope for blind patients to find some kind of cure by improving awareness and specifically perception. I mean, blind sight patients, they have some kind of mind representations, right? So by teaching them how to process stimulation, you will say that they can I mean, maybe right now, no, but they can improve their vision.
Mm-hmm. Yeah, this is quite ambitious in the sense that you are hoping to make something that is unconscious to become conscious. There are different ways in which we can do this. Of course, this is of extreme importance because you have blind patients and you want them to recover. There are basically two different ways of rehabilitating visual field defects. One is just asking them to use strategies, so moving their eyes, but you're not rehabilitating actually. The other way is to keep presenting the stimuli and asking the participant to act on these stimuli, so performing tasks about the stimuli, and you see that they may increase their ability to see something, especially in the central portion of the visual field. The connection between blind sight and the possibility to rehabilitate is not so studied and known. We think we are trying, and with how we should have, but we didn't have enough time to. We were trying to find whether the features of the stimuli, for instance, if you have a simple stimulus may not be enough. You may have, I don't know, faces of people so stimuli that are more arousing for you, more important for you, which can trigger a stronger response in your brain, so making you to change from the unconscious to the conscious state. So there are studies saying that this may happen. We are starting with the myopic patients to run these experiments, and we hope maybe next year I can give you a much better answer about this.
For instance, also the study by using, I don't know,
I didn't hear you.
Now?
No.
Something is happening.
Okay.
Okay, that is better. Okay, I think that maybe, for instance, using another kind of processes like top-down processes, attention or memory, or I mean that's another possibility to increase.
Yeah, but also subcortical structures. Think about emotional faces. If you have fear present fearful faces, you activate the amygdala, you activate all your limbic system, and this can be arousing and increasing the activity of your brain, making your brain behave and works in a higher way, and hopefully to make you become conscious.
Okay. We have another 2 questions. You have time, right?
I'm here.
Okay. 3 questions.
Hi, I have another question. Kind of a follow-up. In your second to last answer, you say something about that there may be another pathway by which the primary visual cortex is not involved in visual awareness. I remember one of your conclusions was that perhaps it's not necessary for visual awareness. And my question is if those pathways may be known or are known, or there's a way to know them.
Yeah. This is saying that primary visual cortex may not be necessary for awareness is a very strong sentence, because of course if I have a damage there I don't see anything, so it is necessary, and it is necessary for normal vision, because if you have a lesion in V1 you are blind. Not totally blind, you may recover, you may have islands of spared vision, but you are blind. What I meant was basically that primary visual cortex may not be the neural correlate of consciousness. So meaning that what happens there may be necessary, but like having your eyes open. If your eyes are closed, you're not conscious, you don't see anything. But you won't say that consciousness is in your eyes, right? So maybe what happens in your primary visual cortex is necessary as it gives the input to other areas, which are those areas, the substrate for consciousness. Maybe the temporal cortex or the parietal cortex. Do I answer your question?
Well, well, the thing is that if I understand correctly, and I'm not sure I did, that might be the case. This also happens with patients with lesions. So there may be perhaps some alternative pathways that do not involve the primary visual cortex.
Absolutely, absolutely. There are, I guess, a dozen of pathways, but the biggest one goes to the superior colliculi, to the pulvinar, to the parietal cortex. And this is, for instance, one reason why one of the areas along this pathway is the area that conceives you to perceive motion, which is MT, in the middle temporal area. Basically, blind sight, if you use moving stimuli, it's more likely for you to see blind sight, which means that maybe you are using this alternative pathway bypassing V1 to reach temporal and parietal cortex. And somehow, not only it conceives you to be better than chancing your behavior, but sometimes also to be conscious of something. Now I responded to your question.
Okay, I kind of get it. Thank you very much.
Okay.
Okay, we have another question.
Hello.
Hello.
Thank you again for your presentation. Very interesting. I was wondering, as you were telling that people can report this this contrast and movement in their blind field. Sorry. I was wondering, like, it kind of reminded me this situation where I don't know, maybe you have heard when somebody else is staring at you, but it's not in your but it's not in your sight field, but you can kind of feel it, you know. I was wondering if is there.
Well, this would have been called blind sight type 2, which is another type of blind sight in which you feel something, you have a sensation, but this sensation is not visual. So the sensation you are describing doesn't have any visual content, right? You have this activation that tells you that something is going on. And this is called blind sight type 2. So you are not visually conscious of something, but you know that something is going on.
That was what I was wondering. And is there some correlation with these conscious areas and the temporary lobe, like similar to the visual experiments you were doing?
I would say that since our brain is mostly performing visual processing and other senses are less represented, they are also less studied. We don't really have studies on olfaction, taste, sounds, as many as we have for the visual cortex. So I actually don't know whether you would perceive the same, you would see the same activations in the brain. I would say that they could be more related to the sense you are using. So if it is acoustic, you would have acoustic areas involved instead of visual areas involved. Even if visual areas are mostly involved in any task, because you also create a mental image, which is typically visual. So vision seems to be our best sense here.
I agree. So, thank you.
Okay, no more questions? Okay, thank you so much, Silvia, for the presentation and for your time, and also for your answers to all these questions. We hope that this is the first time, but not the last one.
Absolutely.
We are looking for some collaborations with you. We are really interested in your field of research, and also we have in our lab research related with that, with perception and awareness, as you know. Okay.
Yeah.
So
yeah.
thank you so much.
Thank you all for having been here.
Okay. See you.
Ciao, ciao.